I'm really excited to be talking with you today about some of the work I've been involved in using nanopore sequencing to classify samples from patients with acute leukemia. So, as some of you probably know, leukemia is cancer of the blood, and it can be divided into two major lineages, being acute myeloid leukemia and acute lymphoblastic leukemia. The important thing to keep in mind is that within these lineages, there are a bunch of different subtypes. And these subtypes both predict patient prognosis and inform clinical decision-making. The focus of much of the work I'll be talking about today is on B-cell acute lymphoblastic leukemia. So I mentioned that there are all these different subtypes and that these subtypes are really important to understand. So how do we actually know what subtype we have? One of the hallmarks of leukemia is the presence of these blast cells. These are immature white blood cells which crowd out healthy blood cells and lead to the disease phenotype that we see in a clinic. And what we can do is examine the genomic profiles of these blast cells and look for signatures which tell us what these different subtypes are. And we understand the most relevant levels to be looking at to be the chromosome level as well as the gene level. So how is this actually done clinically? It's usually done through a series of different assays, one of which being karyotype analysis. This allows clinicians to visualize the different chromosomes and their numbers of copies. And so, for instance, here we can see that the blast cells from this patient contain extra copies of chromosome 21. FISH is another technique which is commonly used to detect gene fusions. I mentioned that we can often see these changes at both the chromosome level as well as the gene level. And FISH is a technique that we use to look at these changes at the gene level. There are a number of other techniques that are also commonly used, including targeted PCR as well as microarray. The primary goal of the work that I've been involved in is to explore whether we can recapitulate these clinical results using a single assay. And so we sequenced about 50 samples from patients with acute leukemia using a Prometheon 2 solo, and we did whole genome sequencing with adaptive sampling. So the basic workflow for this, we would get samples from our biobank. These are samples with known clinical classifications already, and they've been collected in most cases years ago. So we extract our DNA from these samples, we prepare that DNA for sequencing, and we load it onto our flow cell. One important thing to note is that we employ something called adaptive sampling. This allows us to enrich for gene regions of interest. And so in our case, that's about 60 genes that we associate with acute leukemia. And lastly, we have our informatics pipeline. Here we'll do the base calling, alignment, and here we can generate a digital karyotype as well as detect fusions. So what do we actually see when we get our results? What I'm showing you here is the number of reads we're getting across different chromosomes. And so the thing to keep in mind is the importance is to look at the relative number of reads per chromosome. And so based on this, we can infer that there are extra copies of chromosomes 2, 4, etc., etc. So again, we're seeing these changes at the gross chromosome level. If we look a little bit deeper, we can also see these changes at the gene level. And for this particular instance, we see both. And so what I'm showing you here is fusion support for a BCR-ABL1 fusion. The bars in blue are reads which map to both genes. And so these are roughly the first 30 reads that support this gene fusion. Here is another sample that we sequenced. With this, we see a totally normal karyotype. We don't see any differences in chromosome copy number. However, if we look a little bit deeper, we do see several reads supporting a DUX4 IGH gene fusion. Again, the reads which map to both genes are in blue, and our orange and red reads are just wild type reads. So in total, we ran 38 of these samples, and we saw really good consistency with what was determined in a clinical setting. There were 2 inconclusive samples that were not inconsistent with clinical findings, but we couldn't confidently call a classification, mostly due to low blast count. So we were really excited by this retrospective sampling, and so we wanted to understand, what does this look like if we try to do this in a pseudo real-time setting? So recall that these retrospective samples already have a clinical classification. These samples that I'm going to show you now do not yet have that and are active patient cases. And so we basically go through an accelerated biobanking protocol whereby we get a fresh sample and we take it back and run our sequencing in real time. The wet lab side of things takes about 3 hours. And then from there, once we load our sample on the sequencer, we typically are able to confidently call a karyotype within about 15 minutes of the start of sequencing. Fusion detection takes a little bit longer. This typically takes between 3 and 6 hours. And so the important thing to note is that this is all happening in real time. As this data is being generated, it is being analyzed in real time. So from the start, when we collect the sample, to when we can confidently call a classification is usually between 6 and 9 hours. These are examples of some of our real-time or pseudo-real-time cases. They look very similar. Here we see some evidence for aneuploidy. We see different numbers of copies of chromosomes, and we're also able to detect a gene fusion. So of these real-time samples, we had really strong consistency with what was determined clinically in the end. Again, we're getting our results much quicker than the clinical results are coming in, but afterwards we can compare those results, and there was only one sample, again, due to low blast count, that we couldn't confidently call, but that was consistent with clinical findings. All right, so we were really excited by this work. And a few things I want to highlight is that this is a much cheaper alternative to the current standard of care in a clinical setting. Likewise, in a clinical setting, these assays typically take quite a bit longer, and we're able to generate a consistent classification within a day, which is really exciting to us. In addition, because these are long reads, we have the ability to robustly detect the structural variation, which is characteristic of that disease phenotype. So although we're really excited about the data we have generated, there are several things that we're hoping to do, including automating the karyotyping process, as well as optimizing this assay in a cost sense. So that involves downsampling. We are also automatically collecting all of this methylation data with our whole genome sequencing, and we have not yet been able to look at that, but are very excited to check it out. I'm one of many in a very interdisciplinary team, and none of this work would be possible without all of them. So I'd love to thank my collaborators at UNC as well as St. Jude's. Thank you.